With the energy systems, remember, there's three of them. I use the immediate system as the term for that, but that's sometimes referred to as the ATP PCR system. I'll explain here in a bit why I say the immediate system. Then you have the non-oxidative, glycolytic, and then lastly, the oxidative. Oxidative system, oxidative phosphorylation, cellular respiration. These are all kind of terms used, but for the most part, that's the system that keeps us alive as humans. That's what allows for life to happen. The immediate and the glycolytic system are a high power, fatigable energy systems. But when you need ATP, you need it fast. Those are systems that provide it right away and they provide an okay amount to allow for exercise to keep going for a certain amount of time. But the oxidative systems provides a lot of ATP over a long period of time, but it's a slow system. It takes a while to get going on that. Okay, if you work at a slow enough rate, the oxidative system can keep everything going. But unfortunately, that doesn't, there's an intensity relationship with these. And that's what we'll get into once we get through the energy systems. We'll be talking about how intensity and duration all impact what energy system is contributing the most for energy, for ATP resynthesis. Okay, so with this, just remember, 3 energy systems. We're going to go in order from the simplest to the most complex. Also going from the most powerful to the least powerful to the fastest to the slowest. And keep in mind that these energy systems are the distinctive factors that are characteristics of the muscle fiber types we talked about in the last unit. Okay, so just keep going with this and just kind of keep these together, understand these, and just know, kind of keep these all kind of in the forefront as you're learning these systems and keeping them and getting them pieced together. And like I said, don't try and do too much on here. Don't try and overdo it. Just keep it simplified, keep it to the point, and it'll help you to understand this. Getting into the immediate energy system. A title up there too, you see borrowing energy system. Reason why is think of this system as a credit card. So what happens with a credit card is you buy now, you pay later. Put everything on credit and have to pay it back at some point later on. So the immediate system is a lot like that. So it's a system that you'll get a lot of power out of it, but over time you'll have to really pay for it later on. Think of, kind of maybe help you understand this, think of high intensity exercise. Okay, what happens when you do a high intense exercise? Well, can't go that long, but you get a lot of power out of it. But then usually what's happening later on, you're sore. You have to replenish glycogen. You have to replenish a lot of other, well, we'll get into toward the end of the unit, but you have to help replenish hormones and bring everything back down to normal. Okay, with the oxidative system, that is, it's more of a pay-as-you-go type system. Well, you only get out what you can put into it. That's why if you go for a bike ride or a walk or, a really light jog, assuming you're trained at that point, you're not really sore afterwards, so you don't have to really pay back as much. Okay, but the immediate and glycolytic systems are really, you kind of have to, you'll pay it back at some point later on. But the immediate system is definitely that kind of energy system. It's a borrowing system, a credit card system. Okay, you pay now or you buy now and you'll pay it off later on. And so the two parts, reason why I call immediate is because it's, when you look at the two parts right here, there's basically just one step in the reaction. It happens right away. You get the ATP, you don't have to go through a series of reactions to get the ATP. So it happens just like that. So a lot of textbooks you see, a lot of places just call this system is just the ATP PCR or the phosphagen system. Okay, because what's happening in this system is you have ADP plus creatine phosphate via the reaction of CPK, which is creatine phosphokinase, will give you ATP and creatine. So on this reaction right here, it's the phosphagen systems because creatine is basically, well, creatine is a substrate. So here's our first time we're going to talk about a substrate right here. Creatine phosphate, is the substrate itself. And what creatine phosphate does is, creatine in the cell will bring phosphate, will attach on the phosphate, and then it will bring the phosphate in, then via this reaction to be able to put the phosphate on the ADP to give you ATP. Okay, the immediate system right here takes place in the cytoplasm of the cell. So it takes place right there where the cross bridges, it takes place in the M line, so it takes right where the cross bridges are. This system is housed in the M line, and this is where the myosin actin are interacting. Okay, so that's why it's called immediate, because it's just a one step. It brings it in, gets the reaction, and that's it. Okay, so remember, here creatine phosphate is the substrate. And there's always usually plenty of phosphates around, but you start to lose creatine as you start to train. Okay, so as you start to exercise and you do so at a high intensity, you start using up a lot of these creatine in the cell. Well, then you start to lose a substrate. Okay, you start to lose the ability to produce ATP. And so within this reaction, you need creatine phosphate. Even though there's ADPs floating around and there's phosphates around, this reaction is only going to catalyze if you have the creatine and the phosphate together. Because remember, substrates and enzymes go together. And you can see it's creatine phosphokinase. That means that enzyme's only going to work when there's a creatine phosphate molecule present and an ADP present. And I star that because creatine phosphokinase is a rate-limiting enzyme. So that enzyme's either going to speed up or slow down all these reactions. It's only going to occur if you actually need the ATP to keep going through exercise. Okay, so remember, within the phosphagen or the media system, there's two steps. One is you have the phosphagen system, and that is where you have the enzyme creatine phosphokinase, and that catalyzes the reaction of ADP and creatine phosphate to give you ATP and creatine. Okay, so that's the first step. But like I said, in energy systems, you always have the substrate and the enzyme going together. So what happens is as you start to exercise and continue on too long, you start to burn up the creatine phosphate. Therefore, you cannot produce ATP because you don't have the substrate because you've run out of it. So this is where the second step comes in. This is why I call it the media, because there's two parts based upon how hard you're working. For the most part, most people are working really hard, so that's why you need to talk about the second step. So when you start, the problem is you need ATP. And what happens is if you run out of creatine phosphate, you run out of that substrate, you still need ATP. You need another reaction to help catalyze to get the ATP necessary to keep exercise going. And that's the myokinase system. This reaction uses the enzyme myokinase, also known as adenylate kinase, to catalyze the reaction of an ADP and an ADP to give you an ATP and an AMP. That's adenosine monophosphate is the AMP, because I haven't defined that yet, I don't believe. And so this system then will kick on to start giving you ATP, because going back to, and this is where metabolism starts to get a little tricky. And I'm going to do my best to explain this via this video format, but it gets hard, so just kind of we'll go through as slow as I possibly can, and just if you have trouble, go back, re-listen, just keep going back over this next part. So when you're dealing with energy systems, think of the cell as like a river. I always like to kind of just use this example, I guess, because I'm a fisherman. I love to fish out west, and everywhere in the country is like this, but definitely out west, you always see problems with beavers coming in, damming up, flooding up and everything like that. So think of the cell as a nice flowing river, and you want to have all these reactions taking place, and you don't want to build up of anything, because that'll then start to dam up the river. So therefore you'll start to flood up at one point. And so the cell needs to be, you want to operate as flowing as possible. And at rest, everything flows nice and easy. There's no problems. But when you start to exercise, there's where you start to have basically the river starting to dam up in some ways or another. And so during normal operations, you have an ADP, creatine phosphate, via the enzyme, ATP, creatine, you just keep going through this. No problems. But what happens is as you start to exercise, you start to burn up the creatine phosphate and you need ATP. Well, you need to get it from somewhere. And so this is where you start to have a damming up of the cell. So the myokinase system is good for two reasons. One is it gets rid of all those ADPs because if you don't have creatine phosphate present, you cannot produce ATP. So you start to have a buildup of ADP in the cell. The cell never wants to have a buildup of really anything. ATP is good. ATP is basically the currency of the cell, but you don't want to have too much ATP if it's not needed to be used. Just like if I had $1,000, I wouldn't be driving down the road holding that $1,000 outside of the window while I'm driving at 80 miles an hour because it's more likely I'm going to lose it. ATP is costly. You don't want to lose it. So you want to make sure that you have a right amount of ATP, right amount of ADP, and everything else. So what's happening is with this system right here, you run out of creatine phosphate. Well, one, you need to get rid of the ADPs because they're building up too much. And then two, you need ATP. So that's why the myokinase system is nice, is because what takes those ADPs to give you an ATP. Within this system, there's no enzyme within the immediate system that takes an ADP and an AMP to give you an ATP. I know that makes the most sense that you take adenosine diphosphate plus adenosine monophosphate to give you an adenosine triphosphate, but in this system, it doesn't work out that way. So you need to get ATP via the two ADPs. Now, in turn, what happened here is once you get the ATP, then you get an AMP as well. Okay, so now going what I'm saying, things start to dam up. Well, within the myokinase system, it's needed because, one, you get the ATP, but the problem is then you have the buildup of AMP, so you got to get rid of that as well. I'll get into that here in a sec. Okay, so just because you correct one problem, there's still many other problems that keep coming about. Okay, during high-intensity exercise, you're getting rid of the ADPs by producing ATP via the myokinase if you run out of creatine phosphate. However, now you have to deal with the AMP. So the myokinase system is nice because it also works backwards as well. So during exercise, you go 2 ADPs to an ATP and AMP. But when you start to rest and get not needing the ATP, the myokinase will then take the ATP, the AMP, reverse it and make it then 2 ADPs again. So the myokinase system works both ways, which is good because it kind of helps maintain the proper flow of the cell. However, during exercise, when you have the high buildup of AMP, because like I said, reason why we've gotten into the myokinase system is because we've run out of substrate. We've run out of creatine phosphate, but we still need then to keep producing ATP. So that's why we switched to the myokinase, give us the ATP. The problem is now you have to get rid of the AMP. And so what happens then is you go down to this number one, primarily within fast myosins, you start to take the AMP, you've got to clear it out because you don't want a buildup of anything in the cell. That's too much. Too much and too little. Everyone always says like, oh, I don't have enough. That's bad. Well, having too much of something could be just as bad as well. Okay, you have too much in the cell, and that's basically then starting then to overwhelm the cell. So you take the AMP via AMP deaminase, and you get IMP, which is inositol monophosphate, plus NH3, which is ammonia. Okay, so the problem with the buildup of AMP is then you produce this is one of the first byproducts we'll talk about, is ammonia. Okay, ammonia is a caustic chemical, or molecule, I should say. I say chemical because we usually use ammonia to clean bathrooms and toilets and scrub floors and whatnot. Okay, ammonia is bad within the cell because, well, during normal rest or during normal times, we could take the ammonia into the blood, then to the kidneys, then we excrete it out in our urine. But what happens during exercise is you start to funnel blood away from the viscera, from the organs, and then you have it circulating in the blood. And the problem with ammonia is they think it gets into the brain, it causes lower motor neuron dysfunction. So ammonia is one of those chemicals that does cause fatigue. So the problem is when you start to build the AMP is, well, one, you got rid of the ADPs, you got the ATP, but now you have the AMP, which becomes problematic because then you produce ammonia. That's in the fast myosins. Number two, in the slow myosins, you go AMP via non-specific phosphatase to breaks apart then to the adenosine and the phosphate, breaks them apart. So the problem also occurs is when this is going on is, well, one, you're producing a byproduct, which is not good. Ammonia, that's one, that's not good because it causes fatigue. But then number two, on both these reactions, you notice that we start off with the AMP, that is, we start with the adenosine monophosphate, and then both the products are, we don't have the adenosine monophosphate anymore. So that means is we lost the adenosine group. So that means we lost the ability to produce an ATP because we've broken apart that molecule itself. So there's always a cost. That's why we say this is a credit card system, is because you buy now, you pay later. Well, you're getting ATP to keep going, but you're having a lot of these other subsequent byproducts occurring that you're going to have to take care of at a later date. That is, get rid of the ammonia and then somehow get the adenosine back on, and adenosine and the phosphates together to start forming up an AMP, and then to hopefully ADP and ATP. So this is kind of you have to deal with this later on type deal. Now, pros and cons of the systems. Well, the advantages are it's a fast system. Like I said, it occurs in the cytoplasm. So it occurs basically right there, a part of the sarcomere, part where the myosin and actin are interacting. So therefore you have the ATP, boom, right there. One example I always use on this is, if you're hungry, you're not going to drive to the store right away. Right? If you're hungry, for the most part, the first thing you're going to do is you're going to go to the cabinet, go to the fridge, and see what you have in your house. Okay, if I'm hungry, I'm going to go look in the fridge because it's on site. It's located right where I'm at. I'll go there first, check and see if there's food, then I'll go from there. So that's an advantage because ATP is being produced exactly where it's being used. Just like if I'm hungry, I will get the food that is right there in my house right away. Okay, so that's one good advantage. Another advantage is the one-step system. That is, there's one reaction that takes place for both CPK and myokinase. Less chances of failure. I always like to use this example, but the old cars, the Ford Model T, one of the very first automobiles that came out, not that many moving parts in there. So a lot easier to fix than, think of like a Tesla right now that's basically computer operated. Okay, there's a big difference between the two cars. One's easier to fix than the other. And so theoretically, if something goes wrong, there's less chance of failure in those older cars than there's in newer ones, just because of the amount of technology involved with them. So just kind of think of it like that the less steps, less chance of failure. Last is the pump primer. This is byproducts ADP, AMP, ammonia, help activate other metabolic systems, especially the oxidative system. In a lot of ways, ADP concentrations control all of the energy systems. Because you got to keep in mind, like I said, the cell wants to have a nice, smooth operation. So if there's a high amount of ADP being built up in the cell and you need ATP, that's going to kick on the energy systems to start producing ATP. And so a lot of times what you're looking at is the ADP versus ATP ratio. If there's too much ATP, not that much ADP, that's telling the cell that we need to slow down. and vice versa. So ADP really is a big controller for the energy systems. They kick on, especially like I said, the oxidative system. The oxidative system is like, I always use this as, well, us being online is a little bit different, but for the most part, when classes start, no one wants to get up and get going to go to class. So the oxidative system it's like you trying to get up to go to 8 a.m. class on the first Monday of the semester. It takes forever to get up, and trust, for faculty, it's the same thing. It takes sometimes forever to get up, get going. You don't want to go to class. You're tired. You miss summer. The oxidative system is like that. Sometimes to get up and get going, you need somebody to walk in and throw a bucket of water on you and slap you across the face to get you up, get out of bed kind of deal. The oxidative system is kind of like that. It takes the buildup of all these byproducts telling the cell, you need to get up, get going, stop being lazy. So the immediate system helps that out because it starts to have all these byproducts being built up, and it kicks on the other energy systems. Everything with the metabolism is very finely controlled. Okay, and this is just one example of that. Disadvantages, though, it does not resynthesize ATP from ADP or phosphate. From ADP and phosphate. So remember, kind of point number 3 right there. Remember back to neuromuscular when we said if there's a buildup of ADP, a buildup of phosphate, what happens? Too much ADP slows down velocity. Too much phosphate slows down force. So the problem, why do you fatigue out so fast within doing super high intensity type work, like maximal higher intensity, is because the ADP and phosphate concentrations build up too great, so therefore it starts to impact force and velocity, therefore it decreases the power output. So it's hard to maintain a proper cell balance within this because byproducts build up way too fast. That's why when this happens, you'll switch to another energy system, but then you'll slow down. You're limited by concentration of creatine, phosphate, and ATP. There's only so much you could actually store. Creatine phosphate is a ergogenic aid that most everyone's taken. It has shown to be, it's probably it's the only supplement that has unanimous, shows like tremendous amount of support that actually works. Not in everyone, about 20% of the population, but that's pretty good considering how much people have taken it. The reason why it works is because most individuals can't store enough creatine phosphate. We don't store enough, so therefore by taking more in, it helps out, then increase those concentrations and help out them with performance. Okay, so one, you can't get an ATP from ADP and phosphate. You're limited by creatine phosphate concentrations. Remember, all these substrates, they're always going to be a negative because you're going to run out of them. Okay, you're going to run out of creatine phosphate. So it's a negative because you run out, and therefore you can't keep supplying the reactions. Phosphate becomes high, ADP becomes high, we just talked about that. And then you can resynthesize a creatine phosphate, and conversion of AMP and IMP back to ATP are costly. So you could get them all back, it's going to take a lot to get them to go back to the ATP, so it's a very costly process. Glycolysis, referred to non-oxidative, intermediate energy systems. Don't let this picture frighten you, get you scared. It's not as difficult as you're going to think it is. So just kind of keep it simplified here. It'll help out. Okay, remember glycolysis. Break the word apart. Olysis means to break down. Glyco means, glycogen. So we're looking at the breakdown of glycogen, which then when you're breaking down glycogen, you break it down into glucose. So really what glycolysis is the breakdown of glucose. And glycolysis, once again, you can see in the picture, occurs in the cytoplasm. And there's really two fates that the end of glycolysis is going to occur. There's many fates of this one, but I'm kind of showing you where the pathway is going to go first. The two main ones, I should say. So with glycolysis, what it is you're breaking down glucose into pyruvate. Because the end of glycolysis, once glucose has been broken down, you end up with a pyruvate molecule. Well, specifically two. So glucose is a 6-carbon molecule, and then pyruvate is a 3-carbon. It becomes kind of important. We'll talk about that coming up here in a little bit why. So with glycolysis, what's going on is that you're breaking down glucose into 2-pyruvate. And that's it, what glycolysis is at that point. Okay, at that, what other byproducts or other products are given off via glycolysis is you're going to get 2 to 3 ATP. I say 2 to 3 because if you start off with glucose, you'll get 2 ATP. If you start off with glycogen, you'll get 3 ATP at the end. Reason being is because glucose, you can see the picture, or you can see the enzyme 1-glucose, hexokinase, G6P. What happens is as glucose comes into the cell, hexokinase, and make sure you know these enzymes, by the way, as well. As glucose comes into the cell, hexokinase puts a phosphate onto glucose. And it says 6 because it gets placed on the 6th carbon. Okay, you don't need to know that, but just kind of know why the name is what it is. And so what happens is, just like creatine phosphate, glucose 6-phosphate, when the phosphate gets put onto a molecule, it becomes activated. That's why ATP is so powerful. It's because it's that 3rd phosphate gets put on, it activates it, makes it powerful. So the hexokinase 1 is it puts a phosphate on there, becomes glucose 6-phosphate, and then 2, what it does is activates this 1, and then number 2, it traps it into the cell. Because the biggest thing you don't want is for things to go freely across the cell membrane. Because if glucose could just freely flow in, they could just freely flow out, then you'll lose the ability to break it down into ATP. So that's why, that process right there, putting the phosphate on via hexokinase, it takes an ATP to do that. So hence the reason why you get less ATP via glycogen. See, glycogen, I should have said this, but when glycogen gets stored up, it gets phosphates put onto it when it's storing up. So therefore, when you go to break down glycogen, it's already been phosphorylated. So you don't need to actually put a phosphate in because it's already occurring at that point. So what you're getting, that's why you get 2 to 3 ATP depending on what you start with. And then via the whole process, then you get 2 NADHs. The nicotine amide adenine dinucleotide. Remember, we talked about this as a cofactor. Okay, glycolysis will not work. Doesn't matter how much glucose you have, doesn't how much enzymes you have. If there's no NAD present, then glycolysis will not work. Okay, so remember, that's why it becomes important, is those NADs. And you can see in the picture on the left, they collect hydrogens and they keep everything happening, keep everything functioning properly. We'll get into here in a sec why those hydrogens become important. Okay, so when you're looking at glycolysis, don't get nervous on all these pictures, all these names, and all of that. Just go, okay, what is glycolysis? It's the breakdown of glucose into pyruvate. Through the glycolytic process, I get 1 glucose will give me 2 pyruvate, 2 to 3 ATP, 2 NADHs, and leave it at that. Okay, don't go on and beyond that right there because you'll start to confuse yourself. Okay, so just know, glycolysis, I start with glucose, and through the breakdown of glucose, I'll get 2 pyruvate, 2 to 3 ATP, and 2 NADHs. And just keep it that simple for what you need to know for this point right here. And then that pyruvate will have a bunch of different fates, and we'll get into all that right there. But one fate of pyruvate is then hopefully, we'll get into this coming up here in a few minutes, but pyruvate will go into then the Krebs cycle, get converted to acetyl-CoA, and go through then oxidative phosphorylation. Okay, and so you know about hexokinase, what that enzyme does, but one of the most important ones in the glycolysis, and one of the most important ones in the whole entire body, is phosphofructokinase, PFK. And you can see it's the conversion of fructose 6-phosphate to fructose 1,6-disphosphate, or diphosphate, I'm sorry. Okay, you don't necessarily know those terms as much, but know what phosphofructokinase is, and that is the rate limiting enzyme, and it's a committed step. Meaning, phosphofructokinase will either speed up or slow down the process depending on what is needed at the time. If there's a high ADP versus ATP ratio, then it's going to speed up and start giving us ATP. If there's too much ATP and little ADP, then it's going to slow everything down. If there's too much pyruvate, it's going to then slow everything down. If there's not enough pyruvate and it's needed, it'll speed everything up. Okay, if there's not enough NAD then present, then it's going to PFK is going to slow everything down as well. Okay, so PFK really is the biggest rate limiting enzyme in the body, maintaining a proper balance of everything. Remember, we want the cell to be operating like a river, flowing as easy as it possibly can. When you start to have a mismatch, then it's going to slow everything down, and then you need to get things back working properly again. Okay, and PFK is that enzyme that does that. It speeds up, slow down based upon what is present within the cell. And just remember, like I said, all these products we're talking about, they're giving off are good things, but they can become bad if there's too much. Okay, if there's too much pyruvate, well, remember that I said the next step is pyruvate goes to acetylcholine, then in the Krebs cycle. Well, the oxidative system is a very slow system. If it can't handle all that pyruvate coming in at once, they're just going to start to get a backup. There's going to be this backup, so therefore, what's going to happen is it's going to slow everything up before that down. It's going to dam up the entire river. Ideally, you want pyruvate to be produced to slowly go into the Krebs and keep everything going nice and easy that way. So just when you're learning this, just remember that. Just, okay, like how does all these working together right now? First, know like what is glycolysis? It's the breakdown of glucose into 2-pyruvate, 2 to 3 ATP, 2 NADHs. PFK will slow down or speed up the reactions based upon what's present. Okay? And don't try to add more in than what we just talked about at that point right there. Keep in mind, though, that with this system, everything is needed to keep everything working properly. Like you need ADP in the system, you need ATP, you need a proper ratio of that. You need NAD so it can accept the hydrogen and become NADHs. All these things are needed to keep everything going. And those hydrogens become important too, by the way, because that and pyruvate together will give us a molecule we'll talk about here coming up in a little bit in the lecture. So don't forget about those two ones right there. Now the ATP being produced could be used wherever. The NADH will then go to, and you can see in the top right picture, to the electron transport chain for oxidative phosphorylation. Okay, so it'll go to the ETC for that to give us ATP. And you got phosphorylase, the last enzyme I'm going to talk about, but that's just the enzyme that breaks glycogen down to glucose 6-phosphate. So make sure you know those enzymes. Make sure you know PFK, why it's so important, the major part of that. Just make sure you know why that enzyme is so important and how it's going to speed up, slow down based upon what's taking place within the cell. You got advantages and disadvantages. Advantages, so it's a quick reaction. So it occurs in the cytoplasm, just like the immediate system. This one, you can resynthesize ATP by ADP and phosphate. You have the capability to get an ATP from those two. So therefore, it keeps the phosphate and ADP concentrations low. That's all good, helping maintain a proper functioning of the cell. Disadvantages, though, well, just like the immediate system, if a substrate's needed to run it, that's going to be a disadvantage because you'll eventually run out. So this system is dependent upon glycogen and glucose concentrations. It's dependent upon NAD. There's more steps. There's about 12 steps in glycolysis, so there's a greater chance for failure to occur. You only get small amount of ATP, so about 2 to 3 is all you get at the end. So it's not that much more than the immediate system, but not that much more. You got end product waste buildup will eventually lead to a system slowdown. And that's what we'll get into coming up here in a bit, but that main waste product is lactate formation. So I'll just bring this up right now. We'll talk about coming up here in a bit, but lactate forms when there's too much pyruvate being built up. And if you're going to have a buildup of pyruvate, I should have said this a minute ago, but if you have a buildup of one of these products, you're going to have a buildup of the others generally. So if you have too much pyruvate being built up, then you're going to have too much NADH as well. And so you got to make sure, and how do you clear out, make sure the river keeps flowing properly and flowing nice and easy? Well, you clear out the byproducts. And how do you clear out pyruvate and the NADHs? What you do is you take via lactate dehydrogenase is the enzyme. This one will take the hydrogen from the NADH and put it onto a pyruvate to form then lactic acid. And you can see at the bottom down there, the 2 lactic acid, because you got the 2 pyruvate via glucose, put the NADHs onto the 2 pyruvate, gives you lactic acid. That's how you clear it out. Lactate then goes into the bloodstream and then goes to wherever it needs to go to get cleared out. Okay, so that's one of the main. Now, lactic acid doesn't cause fatigue, but problem is lactic acid is being formed because glycolysis is working at such a super high rate. So it's not lactate causing fatigue. It's just you're working at a super high rate. That's causing fatigue, and a byproduct of working at that fatiguing rate is lactate. So lactate does get cleared out. Lactate is actually buffering out the cell. It's taking the hydrogens from NADH because that's where acidity comes from, is from hydrogen buildup. And so lactate is taking the hydrogens basically out of the cell, clearing it out, and then helping the cell keep working, flowing at a nice even rate. So it's beneficial for the proper function of glycolysis. Getting into the oxidative pathway. So this is a slow system. This is a reactive system. Ideally, you want, as pyruvate's being formed from glycolysis, to then comes in to form acetyl-CoA to keep working then properly from there. But a lot of times during exercise, it doesn't work that way. I think this is one area major where big time dam occurs. And you have a big flooding occurring upstream from this point. Is because the oxidase system, although it's the basis of life, like I say, if cellular respiration is the basis of life, it takes a while to get going, and it doesn't work at a fast rate. So, but underneath normal circumstances, what's happening is you're having pyruvate being formed from glycolysis, gets converted into acetyl-CoA, and it starts the entire process right here. And once again, you don't need to know all these. I'll tell you what you need to know. So don't get overwhelmed looking at this picture. But this right here is the Krebs cycle, also known as the citric acid cycle. Part of the reason why it's called that, well, Krebs was the person who found it. Citric acid is because the first reaction that occurs gives you citrate. So that's why citric acid cycle is also referred to as the TCA cycle, the tricarboxylic acid cycle. That's because you produce 3 carbon dioxides during through the Krebs cycle itself. I use Krebs as a term I'll use the most. There's 2 parts to the oxidase system. One is the Krebs cycle, and then the second one is the electron transport chain. Both of these are in the mitochondria, the powerhouse of the cell. Both are vitally important for the production of ATP. One's not more important than the other. Although all the large ATPs being produced in the mitochondria or in ETC, it doesn't mean though that the Krebs cycle is not important. I always like to use this example, but I love music. Some of you those who have talked to me know I love music. I love music. I love going to live shows. And so most of the time when we go to a show, we're always looking at the headliner. That is the main event. So the ETC would be like the main event. Krebs Cycle would be the opening acts. Okay, but the point of the opening act is to get everyone ready, get everyone fired up, lead you into then the opening act. So the Krebs Cycle is basically the opening act of the main event. Get everyone ready to go, get everyone fired up. And then I tell this story, but back in the 60s, there was a group, some of you may have heard, probably most of you have never heard because they were not as popular, called the Monkees. Well, they were touring, and their opening act was Jimi Hendrix. Well, I guarantee most of you know who Jimi Hendrix is. You may not know who the Monkees are, but you know who Jimi Hendrix is. That's a situation where the opening act was more important than the main event. Okay, so that's kind of what the Krebs is. Krebs is the opening act for the ETC. And so this one gets everything going without proper function of the Krebs, then the ETC will not work properly as well. So here's how all of Krebs, and this is all you really need to know. Okay, it looks a lot more daunting than it actually is. And so, read the top part. It says the primary purpose of the Krebs cycle is to break down the compound acetyl-CoA, which, remember, pyruvic acid comes from glycolysis, gets converted into acetyl-CoA. It gets converted, right? Once it gets converted to acetyl-CoA in the mitochondria. Okay, so you're taking acetyl-CoA and you're giving off, and it says, it makes the compounds NADH and FADH. FADH is very similar to NADH, but it stands for flavin adenine dinucleotide. So the purpose of the Krebs cycle is to break down acetyl-CoA into NADH, FADH, and actually you get an ATP as well. And you can see on the left box right there, per pyruvic acid, you'll get 1 NADH from the conversion of pyruvic acid into acetyl-CoA. That's not necessarily part of Krebs at that point. But what you're getting from pyruvic acid is you're getting 1 NADH from pyruvate to acetyl-CoA, and then per turn of Krebs, you're getting 3 NADHs, 1 FADH, and an ATP. Okay, so to go through that again, the purpose of the Krebs cycle is to take acetyl-CoA to give us 3 NADHs, an FADH, and an ATP. That's it. Okay, don't worry about going on any more than anything other than that. It's simply to give us 3 NADHs, an FADH, and ATP. The ATP could be used wherever. The NADH and FADH then are again shipped off to the ETC to be used for ATP production there. Don't try and add any more in here than what we just talked about at that point right there. Okay, now you do get the NADH from pyruvate to acetyl-CoA, so remember that, but that's not actual part of Krebs at that point. That's still in the cytosol at that point. Now, I want you to remember this, though. To get Krebs started, and I talk about this molecule all the time, and students always forget this. But to get the Krebs cycle started, you need acetyl-CoA, but you can see then it's reacting with another molecule, oxaloacetic acid, also oxaloacetate. That's just a common term, oxaloacetate. So to get Krebs cycle going, because remember we said citric acid is the first thing being produced in the Krebs cycle. Well, it's only being produced because you have the reaction of acetyl-CoA plus oxaloacetate. Those two working together gets Krebs cycle started. And I bring this up. Oxaloacetate is vitally important. It's just as important for the cycle as acetyl-CoA. Krebs cycle will not start unless you have both of those present, oxaloacetate and acetyl-CoA. So you need both to get everything started. And students always forget about oxaloacetate all the time. And I keep reminding them, you need to know this, It's so important. And I know it sounds like, why would it be important? Well, the reason why it's important is because, like I said, it keeps the Krebs cycle started and keeps it going. However, this is why it's also important. Oxaloacetate comes from two molecules. It's either produced from carbohydrates or proteins, and that's it. So this is why carbohydrate intake is so important for exercise, is because it keeps the replenishment of oxaloacetate going to keep everything, the Krebs cycle, up and running. You never want to use proteins to get that, because that means you're breaking your body apart for that. So that's why I bring this up so much, is because this is the practical aspect. How do we keep Krebs cycle going? Make sure you have enough carbohydrates. Reason being is because as you break down glucose, you get pyruvate and also oxaloacetate. That pyruvate gets converted to acetyl-CoA. That reacts then with oxaloacetate to keep the Krebs cycle going. No isocitrate dehydrogenase is the main rate-limiting enzyme of Krebs cycle. And then no, also with acetyl-CoA, you get it from carbohydrates, that is via protein. We'll get into this here in a little bit, but comes from fats. Or you can break down proteins into amino acids. So in a lot of ways, when you're looking at the oxidative system here, all three energy systems can supply the acetyl-CoA. I'm sorry, all three macronutrients can supply acetyl-CoA formation. Whereas, like we talked about the immediate system, only creatine phosphates there. We talked about glycolysis, only glucose or glycogen can be used there. However, when you get the oxidative system in the Krebs cycle, well, acetyl-CoA could come from fats, proteins, and or carbohydrates. And once again, don't try and add too much more into what we talked about. Just know what Krebs cycle is just needed to take acetyl-CoA and oxaloacetate form to get the cycle going. And the result is you get 3 NADHs, an FADH, and an ATP. Keep it at that point right there. We always say each turn of Krebs gives us the 3 NADH and the FADHs, because remember, if pyruvate is coming from, if acetyl-CoA is coming from pyruvate, which came from glucose, well, the glucose was broken down into 2 pyruvates. So therefore, 2 pyruvates become 2 acetyl-CoAs. And so per turn, as we say, then you'll get about 10 ATPs are formed. And you see the math right there. So 3 NADHs. Each NADH gives us 2.5 ATP. Each FADH gives us 1.5 ATP. So you get and then plus you get the 1 ATP produced per turn. This says a GTP and ATP are the same thing. So you see per turn of Krebs, you get about 10 ATP. So you already see, if we're getting in the oxidative system, starting off with glucose, we're getting more ATP than we did just glycolysis alone. Second part you get the oxidative system is the electron transport system. This is where oxidative phosphorylation takes place. And so the purpose of the ETS is to use the NADH and FADH to resynthesize ATP. And also the second part, we'll get here in a sec, is to produce water. That's it. All I really want you to know about this. Don't worry about all the extra added stuff. Just know the electron transport system purpose is to produce ATP and water. And the water's on the next slide. Taking the FADH, NADH, as you drop the hydrogens off, because that's what they're doing, is they're carrying the hydrogens to the ETC, drops the hydrogens off, and then from that point, what they're doing then is, that's how you get the water. And then the electrons, when you drop a hydrogen off, it's a 1 electron, 1 proton. This is why it's called electron transport chain or system, is because the electrons then go through a cascade of events and basically then help to produce ATP. That's the simplified form of it. That's all you really know for this. And then cytochrome C oxidase is the major rate limiting enzyme of this system itself. And this is kind of what we just talked about. You can see, and don't worry too much about all this right here, but all you need to know is the NADH and FADH and is where they come in at, drop the hydrogens off. The hydrogens then will help produce water, and then the electrons then help to ATP synthase, which the enzyme helps then to produce ATP. And you can see it down there. It says oxygen serves as the hydrogen acceptor and combines with hydrogen to form the water, keeps ETS going. FADH always produces a little bit less ATP just because of where it enters into those little purple globs right there. Those are proteins. FADH enters in a different spot, so therefore it doesn't travel quite as far, so you won't get as much ATP compared to NADH. Okay, so to keep this simplified, all you need to know is the purpose of the the electron transport system is to take the NADH, FADH to produce ATP and produce water. NADH gives us 2.5 ATP, FADH gives us 1.5 ATP. Just leave it at that. That's all you need to know to keep it as simplified as possible. Now you kind of understand a little bit about those, kind of going back to we produce those NADHs in glycolysis. Well, they need to go, like I said, they need to go to the ETCD for help for energy production. And so we have these hydrogen shuttles. These hydrogen shuttles are basically shuttles that take the NADH formed in glycolysis, transporting them into the mitochondria to give us then the ability to produce ATP. Once again, kind of like glucose, you just can't have these products freely go across the membrane because you could lose the energy potential at some point. And so with these shuttles, first one is the glycerol phosphate shuttle. So to go through them, it says NADH cannot cross membranes, which is good, otherwise it'll leave the cell, not come back. Glycolysis produces 2 NADHs in the cytoplasm, which can be given in the mitochondria, the ETS. Therefore, this represents a loss of energy which the body could use. So you have these hydrogen shuttles, and the glycerol phosphate shuttle, which is shown right here, this is in fast skeletal muscle. The hydrogen is transferred from the NADH to the DHAP to form glycerol phosphate. DHAP, you can see, is dihydroxyacetone phosphate. So NADH goes to dihydroxyacetone phosphate to form glycerol phosphate. I said that the hydrogen goes to that. You form glycerol 3-phosphate, and then that one enters the mitochondria where it gives the hydrogen then to FAD, making it back into DHAP. The FADH enters the ETS and yields 1.5 ATP. So, no, the glycerol phosphate shuttle, the purpose of this is it's in fast skeletal muscle, and it's just a series of steps that's taking the hydrogen from the NADH producing glycolysis, transferring the hydrogen into the mitochondria, and then it gets then converted to FADH, which can be used for energy production. This is the preferred hydrogen shuttle for the body because it works at a super fast rate. Okay, the problem is in this shuttle right here, you're starting with the NADH and you're ending up with an FADH. So therefore, the conversion, you're losing about 1 ATP compared to if we had NADH ending with NADH. However, this is preferred because this works at a super fast rate. So despite the conversion, you lose a little bit of ATP. Overall, you will get more ATP production because the system works at such a fast rate. Okay, you couple that with the malate aspartate shuttle. This is in type 1 fibers, heart fibers, so your slow fibers, endurance type athletes. This one's the one used. This one takes the hydrogen from NADH putting on to oxaloacetate. That's what OAA is right there. Giving us malate. Malate goes into the mitochondria, releases the hydrogen back on the NADH, and then goes to aspartate back again. So this one, you start off with the NADH and you end up with NADH. So there's no energy lost using this shuttle right here. So despite losing energy at 1 ATP from the glycerol phosphate, that one's going to be used because it works at a faster rate than this right here. The glycerol phosphate shuttle, think of it, I always like to say, think of this as like a Ferrari and this one as a moped. Okay, more likely if you drive a Ferrari, you're going to burn through fuel way quicker than you would this one right here. The reason why is you prefer the Ferraris because it goes faster. It's better in the long run than the moped. No offense for moped drivers out there, but we could all agree, I'm sure, that a sports car like a Ferrari is much more desirable than anything else because of how fast you can go. So yeah, you may not get as much fuel per time on this one as opposed to malate aspartate, but because this works at a faster rate, this is preferred because you get more overall in the long run. It goes faster and helps supply those demands a lot better overall in the long run. But both are important because they carry the hydrogens from glycolysis via the NADH into the mitochondria to be used for fuel. So kind of go through this, wrap this part up. So it says resynthesizing ATP from acetyl-CoA derived from carbohydrate. So you got glucose to 2-pyruvate to 2-acetyl-CoA to 2 NADHs. So from glycolysis, you get 2 to 3 ATP, depending on what you use. You get NADH and then 2 NADHs and 2-pyruvate. The 2 NADHs go to the electron transport system. The 2-pyruvate, giving everything working properly and flowing properly, will then go into Krebs. So you see, per pyruvate, you have to double it because there's 2 pyruvates, remember, but for just per pyruvate, you get, from the conversion into acetyl-CoA, you get an NADH. And then per turn of the Krebs cycle, then you'll get 3 NADHs, an FADH, and an ATP. You basically have to double all that, remember, because there's the 2 pyruvate coming from the 1 glucose. And then you go the electron transport system. You got the 2 NADHs from glycolysis outside the mitochondria coming in, 2 FADHs from Krebs, 2 NADHs, pyruvate to acetyl-CoA, 6 NADHs from Krebs inside the mitochondria. Add these all up, you get somewhere in the 30s, and I say somewhere because it's not exact. We don't really know exactly how much, but somewhere in the 30s for ATP. So you can see the combining of the aerobic and anaerobic system gives us way more ATP than just glycolysis or just the anaerobic system by itself. Okay, so this is looking at the two systems coming together. Sometimes you'll hear the term fast glycolysis, slow glycolysis, anaerobic glycolysis, aerobic glycolysis. I don't like those terms. I think those are very misleading and incorrect because glycolysis, by definition, is inside the cytosol or the cytoplasm of the cell. When you're talking about aerobic metabolism, well, you're dealing with the mitochondria. Okay, glycolysis doesn't take place in the mitochondria. And so when I bring this up because I want you to start really distinguishing the different energy systems. Okay, glycolysis is a fast reaction. There's no such thing as slow glycolysis. Once glycolysis is taking place and you form pyruvate goes in the mitochondria, that ceases to be glycolysis at that point. It starts in oxidative phosphorylation. Okay, so once you get in the mitochondria, there is no glycolysis taking place from that point. It's then oxidative phosphorylation. Okay, so when you're dealing with these energy systems, keep them distinct and separate. Okay, I don't like the term fast. I know the book uses fast and slow glycolysis, aerobic, anaerobic glycolysis. Those are very misleading and incorrect terms Okay, when you look at biochemistry and look at the biochemistry books, they don't use those terms so much. Those are more, a lot of times, exercise people have actually started using those to some extent. So, like I said, I don't like those terms. What I use is glycolysis and oxidative phosphorylation. So if I'm looking at this right here, a lot of times what people say this whole entire process I just described would be anaerobic glycolysis, because I went into the mitochondria. It's like, no, you can see I had glycolysis. You can see the first point, and then the next 2 points, Krebs and ETS, those are oxidative system. So we're looking at two separate systems working together. Reason being is because I started off with carbohydrate going all the way through both those two systems. So when we're talking about the benefits, we've talked about the other two, but now with the oxidative system, the advantages is not limited to a single substrate. I can use carbohydrates, I can use fats and proteins, all adding into then the Krebs cycle. We see coming from just carbohydrates, a lot more ATP being resynthesized than just the immediate system or glycolysis alone. This keeps ADP and phosphate low, helps maintain cytoplasmic or NADH concentrations. Okay, so the oxidative system, although it's a reactive slow system, it keeps everything maintained, keeps everything low, and keeps things functioning. Now, problem is it's a slow process. Okay, that the benefit, I should say, is the slow process is nice because it does keep everything kind of flowing properly, like the river flowing properly. However, once exercise starts and you start getting at a higher intensity, that slow process is a disadvantage because things start to back up. You have damming occurring, flooding, and you get problems occurring from there. Okay, this takes place in the mitochondria, away from the action site of the actin and myosin, so it's off-site. So glycolysis, in the immune system, if you're hungry, they're on site. You go to the fridge, see if there's anything there. Hopefully food's there. The oxidative system is, well, there's nothing in the house, so now I got to go. So I got to get in my car. I got to drive to the store. I got to wait in lines. I got to do all this, get the food, drive it home, put it in my house. I got to cook it. Then I could actually consume it. Okay, so it takes a lot longer before you can start using the energy available. It says there's two separate stages, both with several steps. So you have the Krebs and ETS, multiple steps, a lot of chances of failure. Remember, ETS purpose was to produce ATP and produce water. So how does that do it? Well, oxygen is required. So you need O2 available to then have the hydrogens go on to give us H2O. That's why cellular respiration is the basis of human life, because we need water. The water is really the basis of life. And so we need that to keep going. So you need hydrogens and you need O2. That's why when you go to altitude, you become way more glycolytic, is because the available oxygen goes down. You need multiple mechanisms to produce acetyl-CoA. So glycolysis via glucose to pyruvate acetyl-CoA, lipolysis, triglyceride-free fatty acids. You got beta oxidation, which get into that here in a bit. Free fatty acids, acetyl-CoA, proteolysis, protein to amino acids, amino acid deamination to acetyl-CoA, etc. Okay, you need oxaloacetate for this process to work as well. All these are needed. You need acetyl-CoA, you need oxaloacetate, you need a lot of products available to even get the process up and going. And you must have NAD to operate. Without that, both glycolysis and the Krebs cycle will stop functioning. So that's why these cofactors are so important to keep everything up and going, keep everything running. Okay, so those are the advantages, disadvantages. Know for all the energy systems, the advantages and disadvantages of them, because there's pros and cons of all. Okay, and that's why they all work so well together, is because where one fails, that one picks up, and vice versa.